Welcome back, friends. Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding meiosis 2 and its regulation from cell cycle and cell division in detail. So here at the end of the telophase 1, the specific chromosomes still they are under condensed form. So here in order to acquire the decondensation of the chromatin material, now here the specific chromosomes they has to enter into meiosis 2. And here the specific meiosis 2, likewise the meiosis 1, it has various stages. So here the various stages which are a part of the meiosis 2, it may include prophase 2, metaphase 2, anaphase 2 and telophase 2. And here the specific meiosis 2, likewise the normal mitosis, it is an equational division. So here why it is termed as equational division here? Because here there is no change in chromosomal number. So here the chromosomal number it remains constant. So the reason why here meiosis 2 it is an equational division. So here at first we will be studying regarding prophase 2. So here what are the various silent features which are observed during the prophase 2? Is it clear? So let me write it on the board. Meiosis 2. Meiosis 2 is an equational division. Equational division. Because there is no change. There is no change in chromosomal number. There is no change in chromosomal number. And here meiosis 2, likewise the meiosis 1, it has various stages. And here the various stages of the meiosis 2, it may include prophase 2, metaphase 2, anaphase 2 and telophase 2. Is it clear? Meiosis 2 has various stages. Various stages, right? So here the various stages of the meiosis 2, it may include prophase 2, metaphase 2, anaphase 2 and telophase 2. So let me write it here. Prophase 2, prophase 2, metaphase 2, metaphase 2, anaphase 2 and telophase 2. Telophase 2. So at first we will be studying regarding prophase 2. So here after the successful organization of the cytokinesis mechanism, now here the specific chromosomes instantly they enter into prophase 2, which is a part of the meiosis 2. Is it clear? After the successful organization of the cytokinesis mechanism at the end of the telophase 1, now here instantly the specific chromosomes they successfully they enter into prophase 2 of meiosis 2. Is it clear? So let me arrange the things here. So as already mentioned, after the successful organization of the cytokinesis mechanism at the end of the telophase 1, within no time, now here the specific chromosomes they successfully they participate in case of the meiosis 2. So meiosis 2 in general it begins with prophase 2. So here at the very beginning of the prophase 2, here we can observe the compactness of the chromosome. Is it clear? At the very beginning of the prophase 2, here we can observe the compactness of the chromosome, and even the condensation of the specific chromosome it begins at the very beginning of the prophase 2. Is it clear? So let me write it here. At the very beginning of prophase 2, compactness of chromosomes is observed. And here the condensation of chromosomes conjugation of begins during the prophase 2. And here at the end of prophase 2, here we can observe the disintegration of the nuclear envelope. Is it clear? At the end of the prophase 2, again here we can observe the disintegration of or the disappearance of nuclear envelope. Is it clear? At the end of prophase 2, we observe the disintegration of nuclear envelope. So here the disappearance of or the disintegration of the nuclear envelope, it is responsible for the successful separation of the sister chromatids. Is it clear? In order to regulate the separation of the sister chromatids which are a part of the chromosome, here the disappearance of the nuclear envelope, it plays an important role. Is it clear? So here after the successful organization of prophase 2, within no time here we can observe metaphase 2. So here what is metaphase 2 and what exactly happens during the metaphase 2? So let me arrange the things here. At the very beginning of the metaphase 2, here we can observe the movement of the chromosomes towards the equatorial region. So here a specific chromosome which is having two sister chromatids, here it is termed as dyad condition, but whereas the two homologous chromosomes which successfully undergo genetic exchange in order to regulate the crossing over during the meiosis 1, here it is termed as bivalent or tetrad condition. So here the two homologous chromosomes which are having four sister chromatids, here it is termed as bivalent or tetrad condition, but whereas a single chromosome which is having two sister chromatids, here it is termed as dyad condition. Now here the specific chromosomes during the metaphase 2, they are ready to migrate or they are ready to move towards the equatorial region. So here the specific chromosomes, they are ready to move or they are ready to migrate towards the equatorial region by taking the advantage of spindle fibers. So here the specific chromosomes, in order to bind to the spindle fibers, here the chromosomes, they take the advantage of a disc-shaped protein complex termed as kinetochore. So here by taking the advantage of a disc-shaped protein complex termed as kinetochore, here the chromosomes, they are ready to bind to the spindle fibers. Is it clear? So let me write it here. At the very beginning of metaphase 2, we can observe the movement of chromosomes towards the equatorial region. Right? So here the specific chromosomes, in order to move towards the equatorial region, the chromosomes, they has to bind to the spindle fibers. So here for the successful binding of the chromosomes to the spindle fibers, here the chromosomes, they take the advantage of a disc-shaped protein complex termed as kinetochore. In order to regulate the binding of chromosomes to the spindle fibers, chromosomes has to bind to the spindle fibers spindle fibers by the means of a disc-shaped protein complex. Disc-shaped protein complex termed as kinetochore. Is it clear? So here this is regarding metaphase 2. Then after we will be studying regarding anaphase 2. So here what is anaphase 2 and what are the various silent features of anaphase 2? So let me erase the things here. So at the very beginning of the anaphase 2, here we can observe the separation of the sister chromatids toward the spindle poles. So here in order to regulate the separation of the sister chromatids toward the spindle poles, here the centromere it has to undergo splitting. In case of the anaphase 1 of the meiosis 1, we observe segregation of homologous chromosomes or separation of homologous chromosomes, because in case of anaphase 1 of meiosis 1, here splitting of the centromere does not take place. But whereas in case of the anaphase 2 of the meiosis 2, likewise the mitotic cell division, even in case of the anaphase 2 of the meiosis 2, here we can observe the splitting of the centromere. Because of the splitting of the centromere, here the sister chromatids, they undergo successful separation. Is it clear? So let me write it here. During the anaphase 2 of meiosis 2, separation of sister chromatids takes place. Separation of sister chromatids takes place. So here in order to regulate the separation of the sister chromatids, splitting of centromere, it is mandatory here. Is it clear? In case of the anaphase 2 of the meiosis 2, splitting of centromere takes place. But whereas in case of the anaphase 1 of the meiosis 1, splitting of centromere does not take place. So the reason why in case of the anaphase 1 of the meiosis 1, only the segregation of homologous chromosomes takes place. Is it clear? In anaphase 2, splitting of centromere occurs. Splitting of centromere occurs in order to regulate the separation of sister chromatids. Separation of sister chromatids, right? So here when the specific sister chromatids, when they successfully migrate towards the spindle pole, now here the specific sister chromatids, they are ready to migrate towards the spindle pole in accordance with the direction of the centriole. Is it clear? In accordance with the direction of the splitted centriole, the specific sister chromatids, they are ready to migrate towards the spindle pole. Is it clear? So this is regarding anaphase 2 of the meiosis 2. Then after we will be studying regarding telophase 2. So here what are the various silent features which are generally observed during the telophase 2? So let me arrange the things here. So at the very beginning of the telophase 2, here we can observe successful migration of the sister chromatids towards the spindle poles. So here when the sister chromatids, once when they successfully migrate towards the spindle poles, now here the sister chromatids, they are enclosed by the means of nuclear envelope. So here the specific nuclear envelope which successfully disappears at the end of the prophase 2, such specific nuclear envelope again it reappears at the very beginning of the telophase 2. Is it clear? At the very beginning of telophase 2, migration of sister chromatids towards the spindle pole takes place. Towards the spindle pole takes place. So here, after the successful reaching of the sister chromatids to the spindle poles, now here the sister chromatids, they are enclosed by the means of nuclear envelope. Is it clear? Sister chromatids after reaching to the spindle poles, they are enclosed by the means of nuclear envelope, right? Which is further followed by cytokinesis. After the successful enclosure of the sister chromatids by the means of nuclear envelope, here we can observe cytokinesis mechanism. Is it clear? After the successful enclosure of the sister chromatids by the means of nuclear envelope, then after, here we can observe cytokinesis. So here from one single chromosome, here we can observe two haploid daughter cells. So in that case, from the two chromosomes, here we can observe four haploid cells. As I have already mentioned you, at the end of the meiosis 2, here we can observe four haploid cells. Is it clear? So let me write it here. At the end of meiosis 2, we can observe four haploid daughter cells. Is it clear? So here this is regarding meiosis 2 and its regulation from cell cycle and cell division in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.